Welcome to BioBits and Bytes. We are jumping right into the core instruction manual for life today. We're going to be challenging a biological doctrine that has held firm for over 6 decades now. So to set the stage, imagine your DNA is like the universe's most perfect instruction manual, a master recipe book for every cell. Every 3-letter word in there, a codon, has one single non-negotiable meaning. Think of it like a traffic signal that is always red for stop. No exceptions, no interpretation.
Right, and that precision, that lack of ambiguity is the bedrock of, well, all cellular life as we know it. So our mission in this deep dive is kind of twofold. First, we're going to revisit the central dogma, this beautiful system.
Oh, wow.
And then we introduce a recent discovery that really fundamentally changes how we view that precision. We've got some great source material on classical molecular biology, but also these exciting breakthroughs in a group of microbes called Archaea. It turns out they use what we always thought was a flaw ambiguity as a functional feature.
Okay, let's unpack this. We have to start with the foundational rules, right? The central dogma of molecular biology. Francis Crick, 1958. What exactly does this dogma describe, and why is that idea of flow so important?
So the dogma describes the general flow of, well, sequential information in biological systems. At its simplest level, the one we all learn, it's just DNA makes RNA and RNA makes protein.
Transcription, then translation.
Exactly. But the real insight, the piece that holds the most weight, is actually Crick's original definition, which is much more restrictive.
Have you ever wondered why that rule is so strict about what information can't do?
It's strict because of information density. Crick's core, unbreakable idea was that once information gets into a protein, it can't get out again. It can't be used to determine the sequence of another nucleic acid, like DNA or RNA.
So that's the key distinction. It's not just the path forward. It's the absolute impossibility of going backward from a protein.
Yes. That one-way street is what ensures fidelity. It keeps the blueprints safe from, being rewritten by the products.
That makes sense.
And we should say the dogma has seen some modifications over the years, some established exceptions, but none of them have ever violated Crick's core rule.
Right, like reverse transcription. That's the classic example, isn't it?
It is. Retroviruses, like HIV, use an enzyme to go from RNA back into DNA. It's a special transfer, but it's still information flowing between nucleic acids.
It doesn't break that protein barrier.
It keeps the information firmly in that domain. And then you have something really weird like prions.
Yeah, what about prions? When we talk about them, are we even talking about the same kind of information flow?
That's a great question. Let's break it down. Prions are infectious proteins that transmit their shape, their 3D conformation, to other proteins,
Forcing them to misfold.
Exactly. But it's a transfer of shape, not sequence. The amino acid sequence itself remains unchanged. So again, Crick's core idea holds.
Okay, so the law of precision has been pretty solid for 60 years. Now, let's introduce the rule breaker, the Archaea.
This brings us to a microbe called Methanosarcina acetivorans. It's a methane producer. And researchers at UC Berkeley found that this organism allows for, well, a flexible interpretation of its genetic code, a kind of controlled imprecision.
And the specific point where this happens is a familiar one, the UAG codon.
Correct. UAG is the amber stop codon. It's supposed to be the definitive stop end of story signal for protein synthesis in, well, almost everything.
But not in acetivorans. For them, that stop sign sometimes means, what, keep going?
Precisely. The UAG codon gets interpreted in 2 completely different ways from the exact same piece of messenger RNA. It can act as a stop signal, which gives you a shorter, truncated protein, or it acts as a sense codon, inserting the rare 20-second amino acid, something called pyrrolysine.
And that gives you a completely different full-length protein.
A full elongated protein. Yes, a process we call read-through.
So the UAG codon is, I love this phrase from the source, it's like a fork in the road.
It really is. The cell flip-flops, making a choice. But why? Why would evolution introduce that kind of risk?
There has to be a pretty big reward.
A huge metabolic reward. The ability to insert pyrrolysine or pyle is absolutely critical for this microbe. It needs pyle to build the enzymes that let it digest its food source, methylamine.
Okay, and these microbes, acetivorans, they're found in some interesting places. Deep sea vents, sewage, and even the human gut.
Yes, and that's the direct connection to our health. When you eat red meat, for example, your gut flora metabolize the compounds. These archaea use the methylamine. If they don't, other bacteria can turn those byproducts into something called trimethylamine, or TMA.
Which our liver then turns into TMAO, a compound that's been linked to cardiovascular disease.
Exactly right. So these little microbes are actually playing a role in preventing the buildup of a compound linked to heart disease.
And they need this weird, flexible code to do that job.
They do. Which brings us to this key reframing by the researchers that ambiguity is actually a feature. It's not a bug.
I love that. It's not a mistake. It's a design choice. So if it's a choice between stop and go, what's making the decision? It can't be just a random coin flip.
It is absolutely not a coin flip. The old thinking was that any ambiguity would just create, a pool of useless protein junk. But here, the ambiguity is regulated. It's managed by what's called a kinetic competition right at the ribosome.
Okay, let's pause and simplify that part again. So you have two things racing each other to that UAG stop codon.
That's a perfect way to put it. Imagine a high-stakes foot race. In one lane, you have the termination referee, a protein called the release factor.
Its job is to bind to UAG and stop everything.
Halt and release, yes. But in the other lane, you have the specialized delivery truck, the pyrrolysine tRNA. And if that pile tRNA gets to the UAG codon first, it overrides the stop signal and inserts the 22nd amino acid.
Got it. So the whole system is biased by how much pyrrolysine is actually around. If there's a lot of pile in the cell,
Then there are a lot of those pile tRNA delivery trucks zipping around. It becomes much more likely that one of them will win the race against the release factor.
which biases the whole process toward making the full-length methylamine-digesting enzymes.
Precisely.
And if pile is scarce, maybe the microbe hasn't been around its food source for a while.
Then the pile tRNA is rarely seen. The release factor wins the race almost every time, and the UAG codon just goes back to being a normal stop signal.
So the cell avoids wasting energy-making enzymes when it doesn't even have the special building block for them.
It's an incredible integrated sensor and switch. The environment itself provides the feedback loop.
And it's not a small thing for this microbe. The sources say it has something like 200 to 300 genes that use these ambiguous UAG codons.
It's a deeply integrated system.
But wait, that raises a question. If UAG can mean go, doesn't that create chaos in all the other genes where UAG has to mean stop?
That's the critical point. How do they avoid global catastrophe? And the answer is genomic optimization. The researchers found that these pile-using Archaea have significantly fewer UAG codons in their genome overall compared to their relatives.
So evolution has basically cleaned house, removing that codon from places where ambiguity would be a disaster.
It's confined the risk to only where the reward is worth it. It's controlled chaos.
Wow.
And if we connect this natural code expansion, life finding a way to use 22 amino acids, it really validates what humans are trying to do in the field of synthetic genetics.
We're moving beyond nature's toolkit, trying to artificially expand the genetic alphabet itself. This is where xenonucleic acids, or XNAs, come in.
Exactly. XNAs are lab-designed alternatives to DNA and RNA. They can have entirely different chemical backbones, but they can still store information and even evolve.
So what does it actually take to get a cell's machinery to read and copy these completely alien molecules?
It takes a ton of polymerase engineering. Polymerase enzymes, the things that copy DNA, are incredibly specific. They have what's called a steric gate, like a molecular bouncer at a club.
To keep out anything that doesn't look like a standard nucleotide.
Right. So we have to engineer those polymerases, often starting with really high-fidelity ones from Archaea, to accept these new chemistries, like tRNA, HNA, or FANA. It's a huge effort in directed evolution.
So what's the big payoff? Why go to all that trouble?
The key property is biostability, extreme biostability. Natural DNA and RNA, they break down really easily in the body. They get chewed up by enzymes called nucleases.
I see.
But something like an HNA aptamer. An aptamer is just a nucleic acid that binds to a specific target. It can be completely resistant to degradation for over a week.
So for things like diagnostics or drug delivery, they can survive long enough in the body to actually do their job.
Exactly. It makes them transformative.
Okay, here's where it gets really interesting. Let's loop this all back. The natural ambiguity in that little microbe, Methanosarcina, and connect it to human disease.
This is the big leap. The exact problem that Archaea solved, how to manage a tricky stop codon, is directly relevant to a huge class of genetic diseases. We're talking about diseases caused by premature stop codons, or PTCs. They're also called nonsense mutations.
And these account for something like 10% of all inherited genetic diseases. Things like cystic fibrosis, Duchenne muscular dystrophy.
Devastating conditions. A mutation puts a stop signal way too early in a gene. The cell makes a shortened, non-functional protein that usually just gets destroyed.
So the therapeutic goal seems obvious.
Yeah.
You need to somehow convince the ribosome to ignore that premature stop sign.
You want to induce a little bit of controlled, localized imprecision. Make that faulty stop codon just a bit leaky, enough for some ribosomes to read through it.
Because even producing a small fraction of the full-length functional protein can be enough to dramatically reduce the symptom.
It can be. And this is why the Archaea discovery is so important. It gives us a real-world, evolutionarily validated model for exactly this mechanism. It proves that life can not only tolerate but regulate kinetic ambiguity at a stop codon for a specific benefit.
So it provides a blueprint for designing drugs that could maybe tip that kinetic race at the ribosome, turning a flaw into a therapy.
That's the goal, turning imprecision from a bug into a feature, pharmaceutical.
This has been an incredible deep dive. From a 60-year-old biological law to potential cures for cystic fibrosis. Let's do a quick recall for you, the listener.
Okay, first takeaway, the rule. The genetic code was always thought to be completely unambiguously precise during translation.
The break. The Archaea Methanosarcina acetivorans shows that controlled ambiguity is possible. It uses the UAG stop codon to either stop or R to insert the 22nd amino acid, pyrrolysine.
The regulator. And that stop or go choice is not random. It's managed by a kinetic race at the ribosome, a foot race that's biased by how much pyrrolysine is available.
And finally, the payoff. This natural system gives us an evolutionary blueprint for developing therapies that could override premature stop codons in devastating human diseases.
Which leaves us with a really provocative thought for you to mull over. The fact that life can stabilize and actually exploit this ambiguous intermediate stage of code evolution raises a big question. Was the strictly unambiguous genetic code that we see in humans the result of maximum evolutionary perfection? Or was it maybe just a historical accident, a contingency that limited our own potential for flexibility?
A truly fascinating concept to think about as we continue to engineer life's code. Thank you for joining again.